Hi friends, today we will discuss about the nucleus. The nucleus is the most important organelle of the cell. Mostly in eukaryotic cell, the nucleus is the most prominent part of the cell. And nucleus being we can say that repository, or we can say the it holds the very important material, that is what we call it as a deoxyribose nucleic acid, or a DNA, or we can say the hereditary material. This is the most important thing in the cell. So today we will discuss about the nucleus. Its first point is its occurrence. See, in all eukaryotic cell you will find the nucleus. Then in some cases, or in most of the cases, you will find that the nucleus is a uninucleate. That means a single nucleus is present in each and every cell. But there are certain kinds of exceptions. So, for example, liver cells, muscle cells, or platelet producing cells, you will find more than one nuclei, and that's why these such type of cells are called as multinucleate cells. Then even in some cases, for example, in case of RBCs, the nucleus is upset. Now, this is because you see the diameter of the RBCs and the diameter of blood capillaries, that is very small. So, and the to actually these RBCs squeeze through the blood capillaries, and because of that they require, or if the nucleus is present, there will be a kind of a resistance to pass through this capillaries, and that's why they have discarded the nucleus. So it becomes more easy for the RBCs to move through this. That's why these are without nucleus, and that's why they are not called as a red blood cells, but they are called as a RBCs, red blood corpuscles. So that is the reason. Then you will find that this nucleus is the largest organelle. It occupies around 10% of the volume of the cell. So it also suggests that this is the most important thing in the cell, because it controls metabolism, it controls the expression of a gene, various kinds of proteins are synthesized with the instructions of given by the nucleus. So that means nucleus is the most important thing in case of the cell. So this is about the introduction. Now if we see the structure of the nucleus, see, the nucleus is bathing in a cytoplasm, and you will find that the outermost layer of the nucleus is a double walled layer, and that's why it is called as a nuclear envelope. Now here the outer membrane, this one, and the inner membrane. So this outer membrane and inner membrane is continuous at the point of the nuclear pore. So this is a nuclear envelope. Then inside, when we go from outside to inside, then after nuclear envelope, you will find a nucleus, a kind of a semi-solid liquid, that is called as a nucleoplasm. Then in this nucleoplasm, you will find the chromatin. So that is the most important thing, third part of the nucleus. This is the chromatin, and the most innermost part of the nucleus is called as a nucleolus. And one by one, we will see this type of, or we can say the each and every part of the nucleus. So see, these are the components of the nucleus, nuclear envelope, chromatin, the nucleolus, and the nucleoplasm. So see, this is a nuclear envelope. see here, the nuclear envelope is made up of, you can see here, a double membrane structure. Then it has a outer and inner membrane. So outer membrane is on this cytoplasmic side. The inner membrane is on the nucleoplasmic side. So then the space between these two is called as a perinuclear space. So this space in between these two layers is called as a perinuclear space, and this perinuclear space is continuous with the ER lumen. So you can see here, the endoplasmic reticulum is associated with the, it is always associated with the nuclear envelope, and it has been seen that this endoplasmic reticulum is, or it is instrumental in synthesizing the nuclear envelope whenever there is a um, mitosis. So that's why this endoplasmic reticulum is the most important thing for formation of a nuclear envelope. So that's why it is associated. It is always associated with the nuclear membrane. So this is all about the nuclear envelope. And the another important thing in a nuclear envelope is the nuclear lamina. So a fibrous structure of a woven intermediate filaments made up of a protein lamin. So these intermediate filaments that are present on the inner side, they are supporting the inner nuclear membrane, and because of that, or we can say, it maintains the shape of the nucleus. So these lamin proteins, they are of three types, lamin A, lamin B, lamin C. And the function of this lamin protein is to give a kind of a support to the nuclear membrane, as well as it gives a site for attachment of a chromatin. So you can see here this chromatin material that is present in the nucleoplasm, that chromatin material is attached to this lamin protein like this. So if you see, if we enlarge this particular portion, you will find that the inner nuclear membrane. So this is the inner nuclear membrane. On this receptor protein is there. To this receptor protein, this lamin B protein is attached, and to this lamin B protein, lamin A and C are attached. So this lamin A and C is attached to the chromatin. So this is how that the chromatin is anchored to the inner nuclear membrane. So another important thing in a nuclear envelope is a nuclear pore. See, there is a continuous interaction in between the cytoplasm and the nucleoplasm. So there is a continuous exchange of material in between the nucleoplasm and the cytoplasm here. And how this exchange takes place with the help of nuclear pores, and these nuclear pores are present on the nuclear envelope. So 40 to 120 nanometer in diameter, the diameter of this is, it varies from 40 to 120 nanometer in diameter. They are also called as a nuclear pore complex. Why they are called as a nuclear pore complex? Because number of proteins are attached here, or it is made up of number of proteins, different types of structural features. You can see here on the cytoplasmic side, you will find the cytoplasmic filaments like this. Then these cytoplasmic filaments are attached to the cytoplasmic ring, and this cytoplasmic ring on the nuclear side, it is attached to the nucleoplasmic side, nuclear ring. And even this nuclear ring is also having a kind of a basket like structure. This is called as a nuclear basket, and this is present on the nucleoplasmic side. So, and at the center of this is the central pore is present. So through which the number of exchange of certain kinds of molecule, for example, the ribosomes, or a passage of ions, passage of nucleotides, that takes place in between the cytoplasm and the nucleoplasm. So this is the nuclear pore. The functions of a nuclear pore, I have told you that the passage for ions. So it allows the entry of ions from the cytoplasm to the nucleoplasm. It also allows nucleotides from the cytoplasm to the nucleoplasm. Then certain kinds of proteins are also allowed. For example, we know that for a synthesis of mRNA, for a synthesis of a DNA, different kinds of proteins are required, and these proteins are synthesized in the cytoplasm and they are transferred to the nucleoplasm with the help of or via this nuclear pore, and that's why nuclear pores are very important. Even the ribosomal RNA and proteins, they are assembled inside the nucleoplasm and then they are transported to the cytoplasm. So to and fro movement of these particular types of ions, nucleotides and proteins, even ribosomes, that is allowed through this nuclear pore, and that's why this nuclear pore is a very important part of the nuclear envelope. So this is about the nuclear envelope. Then the second and the most important part, that is called as a chromatin. Chromatin is of a two type, euchromatin and heterochromatin. We have studied this also. Euchromatin is a transcriptionally active and it is less dense. So cells actively synthesizing proteins have a large amount of euchromatin. Whereas in case of heterochromatin, you will see that heterochromatin is a transcriptionally inactive, very compact, and most of the time you will find that it cannot synthesize protein because of the compact nature of the this particular heterochromatin. The mRNA polymerase cannot bind to the threads of this DNA and it cannot find a particular proper position in the DNA. It is mechanically very difficult for a mRNA polymerase to bind to the region in a heterochromatin, and that's why it cannot make a mRNA. And from if mRNA is not there, naturally there will be no protein. So not the proteins are not synthesized in a, or we can say from the heterochromatin. So that's why it is transcriptionally considered as a inactive. And as we know that the sperms have a our sperm nucleus contains densely packed chromatin. So sperms, the function of the sperm is that to take up the genetic material to the ovum, and for that purpose it has to move. So the motile sperm should have a very compact body so that it can flow or it can move through the liquid and reach up to the ovum, and for that purpose its each and every part should be compact, and because of that you will find the nucleus is also compact, and that is mostly made up of a heterochromatin, and because of that it becomes more compact and it becomes easy for the sperm to move through the liquid up to the ovum. So that's why mostly the sperm nucleus contains the heterochromatin. Then if we see the chromatin at a molecular level, see in the chromatin is nothing but the DNA and the what we call it as proteins. So these are the special kind of proteins that are or that helps in compacting the DNA molecule. So if we see on the molecular level, you will find that the DNA is associated with this particular type of a histone core, or we can say the group of protein, that is, or a bundle of a protein that is made up of four different types of histone protein. These four different types of histone proteins are H2A, H2B, H3 and H4. Now they are arranged in the form of a octane. So that's why each H2O, uh, each protein, that is, this is represented doubly, and you will find that two molecules of H2O, sorry, the two proteins, two units of H2O, two units of H2B, two units of H3 and two units of H4, that forms a octane, and that is known as a histone core. To this histone core, the DNA is wound on this, and in between the two histone cores, you will find this the DNA thread is linking or wound on a next histone core. So the thread in between these two histone cores, that is called as a linker DNA. So mostly the DNA associated with the nucleosome. So each of this DNA plus this histone core is called as a nucleosome. The DNA associated with this nucleosome is of nearly about 146 base pairs. So we know that ATGC. So if one AT means one base pair. So like that, 146 base pairs are present in this particular, or they are engaged or they are associated with the histone core. Then a linker DNA, again the 146 base pairs are associated with the next histone core. So like that, it's a beads on a string appearance. So these are like a beads on a string appearance. And even now this is the most simplest 10 nanometer structure of the DNA, that is also called as a nucleosome fiber. And then this nucleosome fiber is still more compacted with the help of another protein, histone protein, that is called as H1. So this H1 ties, or we can say it makes this 10 nanometer fiber more compact. So it ties this each and every histone core along with its DNA with the help of this H1 protein, and it is like this. So it forms a nucleosome structure. So even this, actually this H1 is digested, we get the beads on a string appearance. So like that. So that's why the function of H1 is considered as it makes this histone cores in a compact form, or it brings these two histone cores in a compact form and forms such a type of structure, or we can say the 10 nanometer fiber. So this is the 10 nanometer fiber. You can see here each one is the nucleosome, and these nucleosomes are further compacted with the help of a H1 protein. So like that, it forms a kind of a um, uh, string-like structure. This string-like structure still becomes, or we can say it still get compacted, and this compacted fiber is called as a chromatin fiber. So if we enlarge, or we can say if we see that this particular structure is represented by like this, the further condensation of a chromatin, that means if we compact this, then it will appear like this. Now this is a condensed chromatin structure. Then again, if it is further compacted, it will appear like this. So you will see that this particular region of a chromosome, this is a particular region of a chromosome. That means chromatin, when further compacted, and it will form a kind of a chromosome. So chromosomes are nothing but the chromatin, compacted chromatin fibers. And if we see from this, it will, if we see this particular region, or if we enlarge this particular region, it will appear like this. If we enlarge this particular region, it will appear like this. If we enlarge this particular region, it will appear like this. So this is how that these are nothing but the degrees of compaction, and because of that, the highest degree of a compaction of a chromatin, that is achieved in a chromosome. And because of this, or because of the and we know that because of the formation of a chromosome, it becomes easy for during the mitosis to we can say to distribute these particular kind of chromatin material to the cells. So compaction is for proper distribution of chromatin material during the mitosis. So this is the chromatin at a molecular level. Then we will go to the nucleolus. I have told you that nucleolus is the most important thing inside a nucleus, and maybe on the basis of its function of that particular cell, the nucleoli are one or more. So if single is there, it is called as a nucleolus. If more than one is present, then plural is a nucleoli. So it is a dense fibrillar part of the or a region of the nucleus, and the nucleolus contains nucleolar organizers, the part of a chromosome carrying the genes for ribosome synthesis. So uh, this particular region, or we can say this particular the nucleolar region contains that part of a chromatin which have genes for ribosome. So such a type of DNA is called as a ribosomal DNA or rDNA. Now, what is the function of this rDNA? The function of this rDNA is to synthesize the ribosomal RNA, and this ribosomal RNA is involved in formation of ribosomes. So we know that the ribosomes are made up of ribosomal RNA and protein. So this ribosomal RNA, which is synthesized from the rDNA, which is present in the nucleolus, this ribosomal RNA binds with the certain kinds of proteins and forms a ribosomal unit or a subunit, and these ribosomal subunits are they again join to form a ribosome. So this nucleolus region or nucleolar organizer that is specially meant for the ribosomal RNA, and this ribosomal RNA is synthesized from the ribosomal DNA. Now, the function of a nucleolus is to help in synthesizing ribosomes and assembling the ribosomal or RNA subunits. So this dense region in the nucleus indicates cells actively engaged in a protein synthesis, because if the cell is engaged in a protein synthesis, it will require ribosomes for a protein synthesis, and these ribosomes are naturally they are synthesized in a nucleolus. So nucleolus will be a prominent part in a protein synthesizing cells. So that is what nucleolus is. Then Nucleoplasm, it is composed mainly of water with a dissolved salts, enzymes, and organic molecules suspended within it. So naturally, the nucleoplasm is a most important thing, because different kinds of enzymes, different kinds of ions and salts and organic molecules, they are required for synthesis of different kinds of protein. Oh, sorry, not protein actually, different kinds of mRNAs, and these mRNAs are then transported to the cytoplasm, where these in a cytoplasm they synthesize a particular kind of a protein. So nucleoplasm is the most important thing in a nucleus. Nucleolus and chromosomes are surrounded by a nucleoplasm, and it performs the function of a cushion. So it helps in preventing the mechanical damage to the chromosomes or the DNA in a nucleus. So it also provides a medium by which materials such as the enzymes and nucleotides can be transported throughout the nucleus to its various parts. It also protects the nuclear content. So this is all about the nucleoplasm. Then functions of a nucleus. So I have told you that the nucleus is the most important thing in the cell. Why? That is because, see, the first function is regulating gene expression. I have told you that the we know that there are different types of a cells. Now, why we call it as a different types of a cells? Because they perform a different type of functions. For example, a cell from a pancreas, it will perform or it will synthesize a particular kind of a hormone, that is insulin. So that means that particular cell is performing or doing a function of synthesis of a protein. Whereas if we see in a liver cell is actively engaged in detoxification and it is producing a different kind of enzyme, that is we call it as the detoxifying enzyme or a P450 system. So that means different types of enzymes are produced by different types of cells. Now, who governs this, or who decides that this is, you are a pancreatic cell and you will synthesize this particular protein, and you are a liver cell and you will synthesize this particular. Who decides that? It is actually decided by the genes, and this is known as a expression of a gene. So which part of the chromosome, which part of the gene will be active, and which part of the gene will be active in another cell, that is decided by the that particular nucleus, and that's why this is known as regulating the gene expression. So it regulates the gene expression, and thereby it regulates the synthesis of a protein. So this is the first function of the nucleus. Second function is it initiates the cellular reproduction. We have seen that the cells undergo mitosis. They are in a constant, we can say they are in a cell cycle. Mostly the cells are in a cell cycle, and they perform this particular function of division. So for initiating the cellular division, nucleus is the most important thing during the cellular reproduction. Then third one is it stores genetic material necessary for all of these tasks. So whatever the different kinds of proteins that are required, that may be a structural protein, that may be a enzymatic protein, that may be a hormonal protein, all these kinds of proteins are synthesized on the with the help of genes, and these genes are present on the chromatin, or we can say in the DNA, and that DNA is stored in a nucleus, and that's why the nucleus is the most important thing in a cell. Then I have told you already that different kinds of proteins and ribosomes are synthesized in a nucleus with the help of a nucleus. The mostly the ribosomes, which are the central in synthesizing the proteins, they are synthesized in a nucleus. So the nucleus regulates the synthesis of proteins in the cytoplasm through the use of a messenger RNA. Then messenger RNA is transcribed on a DNA segment, and that serves as a template for a protein production that we have seen. And it is produced in a nucleus and travels to the cytoplasm through the nuclear pores. That I have already discussed. That means the mRNA, first of all, it is synthesized or transcribed on a DNA, and then this mRNA is taken to the cytoplasm, and where in a cytoplasm we see that the ribosomes attach to this and they synthesize the protein. That means the mRNA is synthesized in a nucleoplasm and then transported to the cytoplasm via nuclear pore. Similarly, the various kinds of ribosomal RNAs, they are also synthesized in a nucleoplasm, and then they are assembled in the form of a ribosome, and then they are taken to the cytoplasm. Likewise, so this is how that the nucleus functions as a regulatory organelle of the cell, and it decides the reproduction, it decides the gene expression, it decides the protein synthesis, and because of that it is the most important thing in a cell. So this is all about the nucleus and its functions. So thank you very much for patient hearing.